Bismillahirrahmanirrahim. Welcome again, guys. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture, I'm going to talk about T cell development and their maturation. Okay. Now, in the previous videos lecture, we talked about the T cell biology, their overview, and we talked about T cell receptor, and we talked about types of T cell. In types of T cell, we already explained helper T cell, their types, and moreover, we explained cytotoxic T lymphocyte and T regulatory cell, their function, each and everything we talked in detail. So this is another video lecture which is also related to T cell biology, and in this video what we are going to talk about, T cell development and their maturation. So let's start. Okay, friend. So now let me write. Okay. Now, first of all, let me explain the main idea, the brief introduction about the T cell development and their maturation. Then we will explain it with the help of diagram. Okay. What actually we have, we have the T cells that are mainly developed inside the bone marrow. When there is infection occur inside the body, so the T cell will be, activated, and they will be developed and they will be matured. So during disease condition, we can say that the T cell development, or before the disease condition, the T cell development and maturation is the very important process. Okay, once it develops, so then it will perform their function, it will perform their duty or job. Means that they are involved inside the immune system and fight against different kinds of infection and disease. Now, how? I'm not going to explain that how. Let's talk the cell development, the T cell development. Mainly the T cell are developed inside the bone marrow. Okay, so inside the bone marrow there are different immature cells. Through these immature cells, the T cells are developed. Once it develops, so then it will be, migrated from the bone marrow to the bloodstream, and then from the bloodstream to the thymus. And this migration is actually done by different kinds of cytokines, which we call it Heametopoiesis cytokines. Due to this way, the T cell are attracted to the thymus. And inside the thymus, what actually happen, the T cell will get matured. Okay, actually in simple language, if we talked about the maturation of the T cell, thymus is actually, a kind of institute, a kind of department, a kind of school or college, whatever you can say. From there, the T cell should get the degree, or they should get qualification, or they should get knowledge. Okay, inside the thymus there are different sections, which we call that, in simple we call that classes. Okay, in first class, the T cell should be, we can say the receptor of the T cell will be formed. In the second class, the T cell will be trained. In the third class, the T cell will again train, means they will know the self and non-self antigen. And the fourth class, there will be the last test of the T cell, and after that the T cell will get mature, and then it will be, migrated to the bloodstream and perform their function. So actually the T cell which are, passes from each stages, we will explain it later, but these stages are very important for the T cell maturation. The T cell which are graduated, or the T cell which are mature and which are, become functionable and perform their function and perform their duty. How? This knowledge of the T cell is given by the cell which we call that thymocyte. Inside the thymus there are the different kind of cell, like epithelial cell, macrophages, dendritic cell. Each cell are actually called a thymocyte because each cell is present inside the thymus. And each cell are actually, in simple language we can say, these are the teachers which can guide, which can provide knowledge to the T cell, that what you need to do when you have the job inside the immune system. Okay, so this is a little bit introduction, a brief idea about the T cell development and their maturation. Now let's explain it with the help of diagram. Now the diagram which I will draw here, it's not the real diagram, it's just the imaginary and just the, the diagram through which you will just get the idea about the T cell development and their maturation. So first of all, let me draw the bone marrow. that all of you better know that a lot of cells are actually originated from the bone marrow. So similarly, we can say that the T cell are also, originated from the bone marrow. So let's suppose this is the bone marrow. Again, this is not the real diagram. If you want to see the real diagram of the bone marrow, so this is the real diagram, as you can see on the screen. This is the real diagram of the bone marrow. Now let me draw the bone marrow diagram here. This is a rough diagram of the bone marrow. Okay, so let's suppose this is the bone marrow. Okay, inside the bone marrow we have different kinds of the immature cells. So the first cells, let's suppose this is the first cell, we call that uncommunicative pluripotent stem cell. Okay, this cell will be converted into cumulative pluripotent hemopoetic stem cell. So let me write. So this is the cumulative pluripotent hemopoetic stem cell. Okay, after that this cell will be converted into hemopoetic stem cell. This hemopoetic stem cell will then convert it into pluripotent stem cell. After that it will be converted into lymphoid stem cell. Okay, then this lymphoid stem cell will be converted into lymphoblast, or we can say lymphoid. Okay, after that it will be converted to lymphoblast or lymphoid, and finally from this lymphoid or lymphoblast we get the T cell. So let's suppose this is what? This is the T cell. So finally we get the T cell. Remember guys, this T cell is immature. It is not mature T cell. And moreover, they don't have any receptor, any co-receptor molecule. They don't have all these things. But this is just the T cell development, that it is just formed or originated inside this bone marrow. Now once it is formed inside this bone marrow, then the next step that it will be migrated and shifted to this thymus area. So let me draw the thymus. Let me draw the thymus. Okay friend, so now let's suppose this is the thymus. Although this is not the real diagram of the thymus, if you want to see the real diagram of the thymus, so this is actually the real diagram of the thymus. And they have all these portions, as you can see. Okay, inside this thymus there are different regions of the thymus. Let me draw each region. So actually this is the cross section of the thymus. Okay, so we have different portion of the thymus. Okay, so let's suppose these are the layers of the thymus, as you can see. So each layer, let me write the name of each layer. This is what, let me write the name. This is the lymphoid organ, which we call it thymus. Okay, and this first layer is called as subcapsular region. This second layer is called as cortex. This third layer is called as corticomedullary junction. And this last layer is called as medulla. Okay, so for the maturation of the T cell should passes from each layer, which is actually a kind of classes they should attend, through which they will be graduated, or in simple language we can say through which it will be graduated, or in medical term we can say it will be matured. Okay, after that what happened, this thymus will secreting, let's suppose in the middle what we have the blood stream. So let's suppose this is the blood stream. Okay, you know what actually happened? This cortex will secrete some kinds of cytokines, which is actually chemotaxis cytokines. Let's suppose these are the cytokines. Okay, so these cytokines are called as chemotaxis cytokines. Cytokine. It means that they will attract this T cell. You know, there are different types of chemotaxis cytokines. So let me write here. We have, you know, like thymosine, thymotaxine. We have thymic factor, and we have thymopoetine. So these are what? These are the chemotaxis cytokines which are secreted from this thymus. You know, the cells which are matured before, you know, you guys also need to know about the heametopoiesis process as well, because in heametopoiesis process we also talked that how the T cell is developed, how all the cells are actually developed from the bone marrow. We also talked about that. So in heametopoiesis you also get idea about the T cell development. Okay, but here I also explain the T cell development, but you also need to know about heametopoiesis. Now once it forms, so then the cytokines are releases that will attract the T cell. Now the T cell is attracted by each cytokines, which we call a chemotaxis cytokines. Now it will be attracted, and once it attracted, so then it is passes through these layers. First of all, it will be reached to this subcapsular region, and at that region there will be a process that process is called a double negative stage. Okay, now let me write. It have the double negative stage. Now at that stage, what actually happen? There will be a lot of T cells are, you know, migrated. So let's suppose some of the T cell are migrated, as you can see. These are some of the T cell. These are all actually immature T cell. And once it reaches at that specific area, then the thymocyte actually guide these T cell to amplify and to increase their number. After they contact with the thymocyte, so then these T cell will increase their number, as you can see. So now the T cell will increasing their number. So we get a lot of immature T cell. Now this is the first test that they passed, and this is the first knowledge that they obtained. Okay, so once we get a lot of T cell copies, but remember they have the T cell receptor always. No, these are, let's suppose, the T cell receptor they have always. But remember guys, they don't have CD4 and CD8 molecule. Okay, all these T cells have the T cell receptor, this red one representing the T cell receptor, but they don't have CD4 and CD8 molecule. Although they have but a very small in number, which we call that CD negative molecule. So they have all the CD molecules, which we call it CD negative molecule. It means that they have very low number of the CD molecules. Moreover, these cells have, you know, except this CD molecules, they have the TCR alpha beta. Let me write it. They have TCR alpha beta and sigma delta. There are, you know, some types of the genes which are present inside the T cell. So let me draw any one of the genes here. So let's suppose, let's suppose this is the DNA molecule. You know, this DNA, as you can see, there is also a question arises in our mind that how actually each TCR are formed. Although before that there is no TCR, as I told you, but how this TCR are formed inside this first stage of the double negative stage, how this formed in this subcapsular region, it will be formed that this thymocyte inside the this region, there are the thymocyte, this green one representing the thymocyte. The thymocyte will secreting all these the chemotaxis factor. Actually, this chemotaxis factor will affect on the DNA, as you can see. They will affect on the DNA of the T cell. The T cell DNA will transcribe and translate it and produce two types of the what we can say molecule, which we call that protein molecule. We call that RAG1 and RAG2, recombinase DNA molecule. These RAG1 and RAG2 will affect on the same cell, means they have autocrine function, and when they affect on the same cell, then this cell, these protein molecule actually rearrange the DNA to transcribe and translate and produce the TCR, moreover CD molecule, but in a small number. After that we get the TCR, as we can see on the surface. So this is how the TCR are formed, and a small number of CD molecule are formed, which we call that CD negative. And this stage is called a double negative stage, which will be occur at subcapsular region. Once all this process is done, then all these cells will be shifted to the next region, which we call that cortex region. What happened at this cortex region? There will be, again thymocyte, which we call a dendritic cell or any antigen presenting cell, let's suppose. So let's suppose this is another stage. Okay, so let's suppose this is what? This is the dendritic cell or thymocyte, let's suppose. They actually presenting a kind of antigen to their receptor, as you can see. Moreover, on the other hand, each T cell are also shifted, and let's suppose I will draw just one T cell here. They will, what actually they will do? They will recognize this one antigen through their TCR receptor, as you can see. Once they recognize it, once they recognize that, so after that what happen? They will also, generating or producing another co-receptor molecule, which we call that CD4, and moreover they also, expressing another co-receptor molecule, which we call that CD8. So let's suppose this black one is CD4 and this green one is CD8. Once it's expressed, so then what happen with this means this one cell, they will also recognize the co-receptor of this T cell with the help of their own co-receptor, which we call that MHC. So if this is the CD4, so they will recognize, this CD4 will be recognized by MHC2 molecule. And if this is CD8, so it will be recognized by the MHC1 molecule. So now let me once again tell you that what actually going on in the second stage. The second stage is called as, I forget to write, the second stage is called as double negative stage, double positive stage. Okay, so in this stage, once again let me explain. In this stage we have the thymocyte, which is the dendritic cell or antigen presenting cell. Okay, they will have the MHC1 as well as MHC2 co-receptor or molecules, and moreover they have also a T cell receptor, as you can see. They have moreover on the other hand we have the T cell which have the T cell receptor and CD molecules, but in deactivated form. Now once it's shifted to this cortex region, so then what actually happen? The dendritic cell or the antigen presenting cell will guide the T cell that this CD4 molecule will be attracted to MHC2 and this CD8 molecule will be attracted to MHC1. So they will just guide the CD molecules of the T cell. So here the CD molecule will be guided, and this is called as double positive stage, because here we get the CD4 positive as well as CD8 positive. It means that the CD molecules are matured. at this second stage. Okay, and moreover, once it is recognized, so then it will be selected and shifted to the next stage, which we call a corticomedullary junction. If it doesn't recognize this one CD molecule, so then this cell will undergo apoptosis, means they will secrete some kinds of cytokines, which we call that FAS. This FAS will ultimately kill this T cell. If this T cell doesn't recognize, then okay, let's suppose it doesn't, MHC1 doesn't recognize CD8 as well as MHC2 doesn't recognize CD4, so then this cell will undergo apoptosis. So all this phenomena is called as positive selection. What we call it? We call that positive selection. Okay. Now after that, this cell will be shifted to the next stage. Inside the next stage, again we have this MHC molecule. So let me draw. So again, this is what? This is the dendrite. This is, let's suppose, the thymocyte. On the other hand, we have the T cell. Okay. Now the next test of the T cell inside this one layer, what actually happened? This is the third stage. What happened in this case? In this case, they will, take the test of this T cell. Means they will see that either the T cell is matured or no, either the T cell is recognizing the self-antigen or the non-self antigen. They actually, presented sometime the self-antigen, let's suppose, to their receptor. Okay. So now, what actually happened at this stage? That this black one, as you can see, these are what? These are the CD4, and these are the CD8 positive. Okay, what happened in this case? In this case, the T cell will, this macrophages or this antigen presenting cell will just identify the T cell. They will present a kind of antigen, or which will be the body part, or which will be no foreign antigen. If this is, let's suppose, the body antigen or self antigen, we call that, let's suppose, if it is presenting the self antigen to the T cell, so then what actually happen? If they recognize this self antigen, so then it will be killed. Why? Because in the future, this cell, once released to the bloodstream, so then they will recognizing, and they will fight against the body tissue. Means they will fight against the self antigen, and self antigen is actually the part of the body. So if they are against to the self antigen, so then all the tissue will be damaged, and this disease is called as autoimmune disease. So this is wrong. Okay, so if they recognize this self antigen, so this cell will be also killed. On the other hand, if this peptide, self peptide, is not recognized, so then it will be selected and shifted to the next stage. On the other hand, if we say, if this antigen presenting cell presenting the non-self antigen, and if still it not recognize the non-self antigen, again this is rejected. Why? Because in the future it will be not recognizing the non-self antigen, and it will be harmful to the body. And moreover, this cell will be also rejected. Let's suppose normally what actually happened. They recognize the non-self antigen of the this which is presented by this non-self antigen, which is presented by this antigen presenting cell. This whole process is called as negative selection. Negative selection. Okay. After that, this cell will be passed to the next stage, which we call that medulla section. In medulla section, what actually happened? This cell will further more trained. Means that, let's suppose, if this is the, let's suppose, the T cell, and at opposite side, what we have the antigen presenting cell. Okay. So let's suppose these are the thymocyte, and in the mid we have the T cell. No, this is the last test that they should pass. So from this last stage, once they are, trained, so then it will be, shifted and perform their function inside this bloodstream. In this last stage, what actually happen? At one end of this T cell thymocyte, they have the MHC1 molecule, and this MHC1 molecule actually having the antigen. Similarly, they have also this cell have also the MHC molecule, and they have also what? Antigen. Okay, let's suppose this thymocyte have MHC, and we call that MHC1 molecule. MHC1 molecule, and they have MHC molecule, which we call that MHC2 molecule. MHC2 molecule. Okay, now they have the T cell receptor, as you can see. What happen if this cell, the T cell, if this T cell recognizing this MHC1 molecule, as you can see, so then what actually happen? The gene which is present inside this cell will be, transcribed and translated, and it will produce more and more CD8 molecule. So then it will be, converted into CD8 molecule, and moreover, the CD4 molecule are degraded. It means that upregulation of CD8 molecule will occur and downregulation of CD4 molecule will occur at that time when the T cell recognize MHC1 molecule. Okay, and once it recognizes, so then it will mature into CD8 cell. So after that, it will be mature into CD8 positive cell and having, all the receptors. So let's suppose this is what? This is the core receptor, which is the CD8, and moreover they have also the TCR receptor and other receptor as well. And then it is shifted into the bloodstream, and then this CD8 become cytotoxic T cell. And once it becomes or differentiate into cytotoxic T cell, it performs their function against infected cell, those cells which are infected to bacteria or viruses. So then this cell will perform their function. I already explained the cytotoxic T lymphocyte in detail. You can go back and watch about the cytotoxic T cell. Okay. So this is about the cytotoxic T cell, that how it develops. After that, once, sometime if this cell, let's suppose at the second time, if this cell, recognizing this one, MHC2 molecule. So after that, what happened? The genes are transcribed and translated and produce a large number of CD4 molecule, CD4 positive molecule at their surface. And the gene are transcribed and translated, due to which the CD8 molecule are degraded. And once CD8 molecule are degraded, CD4 molecule are, increase in a large number around the cell. So then what happen? We, this phenomenon is called as downregulation of CD8 molecule and upregulation of CD4 molecule. After that, we get this one cell become a CD4. So then this cell become a CD4 positive. And it will be, accepted, and then it will shift it to the bloodstream and perform their function. Means it will be converted into helper T cell. In helper T cell, it will further convert into helper T cell 1, helper T cell 2, helper T cell 17, follicular helper T cell, and T regulatory cell. Okay, so then it will be further converted into that types of the cell. So I already explained the helper T cell in detail. Their types also, I explained it in detail. You can go back and watch the helper T cell function and cytokines and each and everything. You will get idea about the helper T cell, that how actually they perform their function. So this is actually how they can get mature. Remember guys, if the same process are not done, if they are failed, means if they doesn't recognize as well as this MHC1, moreover they doesn't recognize MHC2, so then they will again die. They will secrete fast molecule, these one cells, and due to this fast molecule the cell will die, undergo apoptosis, the cell will die. Second way, what happened? If this cell recognizing both MHC1 as well as MHC2, again this cell will die. They will recognize any one of them. Then they will be accepted and passed and shifted to the bloodstream and perform their function. Okay, so this is all about the T cell maturation and the T cell development. Okay, so we talked in simple language. What we can say this thymus is actually a kind of institute or department, and in medical words we can say the thymus is that region where the T cell are mature. Now, if the thymus, this section, as we can see, the different section of the T regulator, this thymus, we call that different section of the thymus, and this, let's suppose in the simple language, we call that classes of the T cell. So when the T cell are present at that class, let's suppose at that stage, so then what actually they will be divided and increase their receptor, but it is immature receptor. After that they will be shifted to the next class. In the next class they will be their CD molecule will be activated, and after that they are recognizing the antigen, and after that they will be finally converted into a CD4 or CD8 molar cell, and after that then they will perform their function. So this is actually a kind of class, this is a kind of institute, and this section is a kind of classes, and this T cell is a kind of students which will perform their function in the future in the immune system. And these cells, these thymocytes, are actually the teacher which give knowledge to the T cell that what you need to do, and the stages which they are passes, actually these stages are actually the tests which are passed by each T cell. Once they pass these tests, so then it will shift it to the bloodstream and perform their function. So there is also a question arise that each maturation stages all these stages will almost take 3 weeks. So that's all about the T cell maturation. So that's all about the T cell maturation and their development in detail. I hope you guys get idea about that. If still you have any kind of question in your mind, you can write your question in the comment. I will try my best to give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and definitely subscribe the channel to get more interesting videos like that. Thank you so much for watching.